I'd just like to summarize some of the material from the week 10 lecture where we looked at another tumor suppressor gene, p53. So p53, like retinoblastoma, functions to suppress cell proliferation. Okay, so it's like the brakes. If we go to week 14 where I showed that video discussing oncogenes and tumor suppressor genes, recall how they thought of tumor suppressor genes as the brakes of a car. So a functional p53 is like the brake, and if you inactivate p53, then you lose the brake. So if you lose p53, you lose that negative regulation of cell proliferation. So if you lose p53, you get the increased growth characteristic. And this is different from oncogenes, whereas oncogenes drive cell growth and cell proliferation, and you can have a mutation in an oncogene that makes it more active and that drives proliferation stronger. With the tumor suppressors, if you got this protein that's acting as a brake, if you knock out one copy of the protein, the normal wild-type gene will act as a brake still, so you don't see a particularly strong phenotype. Now, there's a bit of a quirk with the way that p53 functions, and it doesn't follow the normal Knudson sort of idea of a two-hit hypothesis, but we'll look at that in the next few slides. So p53 mutations have been picked up in a bunch of cancers, and actually it's a whole bunch more cancers. So just like the oncoproteins, people have looked in various cancers and picked up degrees of mutation in the p53 gene. Now, typically when we think of tumor suppressors, as I was saying earlier, we think that you need to knock out both copies of the gene. So you need to inactivate homozygously before you see a strong phenotype. And like other tumor suppressor genes, it acts as a negative regulator of proliferation. And now when we looked at retinoblastoma as a tumor suppressor, it has an effect on development. So if you've got retinoblastoma mutations in the embryo, you have effectively newborn infants born with cancer, and it's a cancer of the eye. Now, with p53 mutations, there is normal embryo development, okay, but during the growth of the lifespan of in this case it's the model mice, you tend to see a lot more you see less survival over time. Okay, so p53 isn't required for development, but it's required for survival of the adult. Okay, so p53 is responsive during, and is required for survival of newborn mice going into adulthood. Now, there are the various so like I said, p53 doesn't seem to obey Knudson's scheme of two-hit hypothesis for but when you look at it in more detail, it actually does. Okay, now the complication here is that let me get to the next slide. Yeah, so this is what we discussed. Now, p53 doesn't work as a monomer. It works as a tetramer. Okay, so if you've got two copies of the gene giving rise to two proteins, the mutated gene gives rise to a mutated protein. Okay, because this functions. So typically, if it worked as a monomer, you'd have a completely functional wild type, and this completely functional wild type would suppress the mutated form, because the breaks would still be on, and the failed break wouldn't matter so much. But because it acts as a tetramer, you get a range of, you get pairing of the proteins. It doesn't matter whether the gene was mutated or whether the gene is wild type. The two copies of the gene give rise to proteins, and these proteins come together to make the tetramer, okay, or the homotetramer. Okay, it's the same protein 4 times. And in this scheme, you can quite clearly see that there's 15 out of 16 of the combinations will contain the wild type, and 15 out of 16 will contain the mutant. Okay, there's only a small proportion that is only wild type and a small proportion that's only mutant. So this means that the mutant version of the gene, in this case the red cartoon representation here, the mutant form of the gene affects a high percentage of the different tetramers. Okay, so in this sense, it's an extra layer of complexity over the normal sort of phenotype you'd see if it acted as a monomer. So let me skip ahead here. Now, p53 is not required for development, but it is required for cell survival to various insults, okay, to various sort of environmental factors. Now, when the cell is exposed to various treatments, the levels of p53 increase greatly. Okay, and it's that increase in the break that is helpful for the cell to survive. Okay, now p53 is a transcription factor, so when levels go up, it will turn on a bunch of genes. But typically, in a normal cell, the levels of p53 are low because it's made and broken down quickly. Okay, so the messenger RNA is not stable, so even though it's constantly being manufactured, it's constantly being broken down. But when the cell is confronted with an environmental nasty, somehow the levels of p53 are not broken down, so the constant turnover leads to a quick rise in p53, and then the p53 can step in and affect the phenotype of the cell and make the cell give the cell a chance to survive. So and we'll look at how that operates. So I don't want to go into too much detail here in this summary slide, but yes, so like I'm saying, the levels of p53 increase in response to various physiological signals, and there are typically p53 exists in a low steady state, constantly turning over, but low levels, and in response to these various external stimuli, the degradation of p53 is blocked, and therefore the levels of p53 increase because it's constantly being made. In other words, we're not turning on the gene to a high level in response, we're actually stopping breakdown in response to the external stimuli, and that leads to an increase in the levels of p53. Now, increased levels of p53 are seen when you treat cells with X-rays, ultraviolet, certain compounds that, clinical drugs that damage DNA, and a whole bunch of other factors. P53 levels also increase when levels of the E2F transcription factor are high, and when a few other cellular cues are triggered. And not all of these it's not understood how some of these cues lead to an increase in p53, but it's been documented in the literature. So effectively, we have p53 here as a central node that is able to perceive a lack of nucleotides, ultraviolet radiation, things that would affect DNA integrity. Now, when the DNA is is damaged, the levels of p53 increase because it's not broken down, and as levels of p53 increase, it leads to drive cell cycle arrest and DNA repair, and it gives the cell a chance to either repair damage or go into cell death, so that the damaged cells are removed from the system. So p53 helps make this decision that the cell has to make when the DNA is damaged, of whether the cell can pause and repair damage, or whether the damage is too much and therefore the cell instigates apoptosis and programmed cell death. So I'd like to give a little bit of background of how p53 actually causes these phenotypes, its effect. Now, if we think back to the previous lecture and previous sort of undergraduate study, you know that the cell cycle has various checkpoints. One of them is late G1, and we know that the cyclin-dependent kinases are important to phosphorylate retinoblastoma and other things to allow cells to progress through the checkpoint. Now, what also exists is that there are inhibitors of these kinases. Okay, so there's a bunch of factors that inhibit these cyclin-dependent kinases, and if you inhibit the kinases, you inhibit cell cycle progression. Okay, and p21 is one of the factors that inhibit the cyclin-dependent kinases and therefore inhibit progression in the cell cycle. So it probably comes as no surprise to you that p53 is going to regulate p21. Okay, so when p53 goes up, p21 is expressed. p21 inhibits the kinases. When you inhibit the kinases, you don't allow the cells to pass the checkpoint, and therefore you stop cells from growing. Okay, so when the cell is damaged, the increase in p53 levels pause the cell cycle and stop cells from growing. This is the brake function of p53. Okay. And when you look at various tumor samples, it's clear that many human tumors have mutated p53, and without p53, the cell can't stop the cell cycle progression, and you get damaged cells growing and becoming more unregulated and growing more extensively. Here's a quick slide showing that in a normal wild-type cell, when you treat cells, when the p53 is intact, you get the brake on cell growth, and you get the cells stopping from cell growth and going into some phenotypes such as apoptosis. When p53 is mutated and you treat cells with some kind of treatment that's harsh to the cells don't die, but they lose regulation in growth and they become malignant. Now, recall that p53 was not required for normal embryo development, because during normal embryo development, the cells are protected and not being insulted, and they don't need. So p53 is not part of the story. But during lifespan, cells come in contact with levels of stress, whether it be oxidative stress or ultraviolet stress or other stresses. And when in a normal cell, the cells are confronted with stresses that damage the DNA, p53 breaks the cell cycle and stops the cells. Okay, and that's why it's a tumor suppressor. So damaged DNA signals to stabilize p53. So p53 is not broken down upon, and therefore its levels build up. And there's a mechanism that relates DNA damage to p53 stabilization. Other things cause p53 stabilization as well, which I'm not going to go into in this summary. And so effectively, p53 is acting as a a circuit that converts, insult into a restriction of growth. Now, normally, I've been saying to you that p53 is degraded as it's made, so its levels don't build up. And the mechanism by which it's degraded is that it's tagged for degradation. It's tagged, and then it becomes ubiquitinated, and then it's broken down by the proteasome system. So now the degradation of p53, which is the normal wild-type state, in a normal healthy cell, p53 has been broken down. It's been broken down because of MDM2, which is a protein which tags it for degradation. So there's a little diagram here showing here. Here's p53, and p53 is a transcription factor. Typically, it's present in low levels in the cell because it's being broken down all the time. Okay, and what's happening? p53 is activating genes at a low level, and one of those genes is the MDM2 gene. Okay, now I've just said to you that MDM2 is one of the genes that tags p53 for degradation. So as MDM2 levels build up, it feeds back to tag the p53 for breakdown. So in this system here, as p53 levels start to increase, the MDM2 tags it to break down. So it's a feedback loop, keeping the levels low. Now there are various things that can happen to the cell that interrupt this scheme whereby p53 is not broken down. And DNA damage is one of the key factors which will interfere with this process, leading to activation of DNA damage leads to activation of other factors which phosphorylate the MDM2, which then interfere with the breakdown of p53. And therefore, p53 levels can build up. Now, we talked a little bit previously about this idea of various growth factors feeding into control the cell cycle through retinoblastoma. Now, it turns out that p53 is responsive to the levels of retinoblastoma. So this tumor suppressor here is monitoring what's going on here. So if p53 is not functioning in the cell properly, p53 is unable to inhibit E2F. Recall from the last lecture that p53 is an inhibitor of this transcription factor, and when p53 is phosphorylated, the transcription factor becomes released and becomes active and turns on genes for DNA synthesis. In a situation where p53 is eliminated from the cell somehow, then E2F levels rise because it's not being inhibited. E2F turns on a particular gene. Now, this gene is an inhibitor of the MDM2. Now, remember, MDM2 targets p53 for degradation. If you inhibit MDM2, because this is active and therefore this is active and therefore this becomes inhibited, then p53 is not broken down, and therefore the cell is able to then, p53 is then able to turn on genes which push the cell into apoptosis. So effectively, p53 is wired in to the system at the G1 checkpoint of the cell, and by monitoring what's going on with the retinoblastoma, p53 can push the cells into apoptosis if retinoblastoma is inactivated. Okay, so effectively we have a system here, as I've just described. Now, how does p53 work? p53 is in itself a transcription factor, and it recognizes a particular motif that's been identified. So this is the repeat motif that if this sequence exists upstream in a promoter, upstream of a coding sequence in a promoter, this sequence will bind p53. So it's very easy to search in databases to see which genes contain this motif. And if you look in detail at which genes contain this motif, there's a bunch of genes. I'll just fast forward here. Yeah, so there's a bunch of genes here which are induced by the p53 because they have that binding motif. And these are all genes involved with DNA repair. Okay, so it's not DNA synthesis gene set that's turned on, it's a DNA repair gene set that's turned on. So when levels of p53 increase, then the cell pauses, the cell cycle can pause, and p53 drives the expression of genes that are required for DNA synthesis. And if the DNA is repaired, then the cell can progress. Now, one of the genes that is activated by p53 is this gene p21. And recall that p21 is an inhibitor of the cycle-independent kinases. So when p53 goes up, the p21 goes up, p21 goes up, cycle-independent kinases are inhibited, and therefore checkpoints can't be crossed, and therefore the cell cycle pauses. Okay, and then once the levels of p53 return to normal, cell cycle can progress again. So p53 is really involved with sensing DNA damage in cells and causing cell cycle arrest and helping the cell turn on functions that are important for DNA repair. So in this sense, p53 is referred to as the guardian of the genome because it plays such an important role. But it's also a bit of a weak spot in the cell as well because it makes this decision. It's not a system that is redundant. There aren't other proteins in the cell that can fill in for p53. So if p53 is mutated, it's a big consequence to the cell because you're losing that cell break. Alright, I'll cut short there, and hopefully that was useful as an overview of the lecture. Again, don't forget, for all of these lectures on oncogenes and tumor suppressors and things, I've put those chapters into the on views from Weinberg, which cover this in more detail if you want some more detail. Okay, thank you.